Hello.
Hello.
How are you?
Pretty good.
All right. I don't know if Mehul can hear me.
Yeah, I can hear you.
There we go.
Am I audible now?
Yeah, you're audible.
Okay,
All right.
great. I don't know what was the issue. Yeah,
Yeah.
I changed the browser. It worked.
Okay.
So yeah. Hi
Oh,
everyone.
So.
Yeah.
Yeah.
So I have been going through my previous work. What I was up to was previously dynamic hypergraphs and these metrics. Like I actually tried a bunch of metrics. Now I'm like now I'm forgetting a little bit of what I did last time, like what metrics meant what and in what context it can be relevant. So I'm still on it. I don't have any updates on that. Like it's more of like I'm refreshing my memory and setting up the environment.
Okay.
But I wanted to attend.
All right. So yeah. Do you think you can give a presentation out on that at some point soon, or?
Yeah.
Because I think that would be good
Definitely.
to go back and refresh what happened last year and give some more thoughts on it. Would be good.
Yeah, I think that will be good. Can I do it next week on 3rd of February?
Yeah, I think so. I think you can do it next week. We don't have anything else scheduled, so.
Yeah, it is like after like 2 weeks, after 14 days, so it's like 3rd February.
Yeah. Oh, okay. So 2 weeks.
Yeah, so this week I'm traveling and I might miss on it, so don't want to promise like next week. So that is why I'm asking.
Okay. Oh, that's fine. Yeah, I think so. That's good. All right.
Yeah, that is it from my side.
All right, that's it. How about Susan? Did you have any more thoughts about the tensegrity stuff?
No, I've been building another desk here so that two people can work in this space. There is construction material behind me there.
Yeah.
So I mostly got it done. I just have to do the wiring.
Oh, okay.
You have to add a new plug-in for it. But yeah, so I might be in a different spot next week. It'll be very close to this spot, but anyway, now I sort of took a week off to try to straighten things up around here.
Yeah.
Maybe it's going to take 2 or 3 weeks, maybe.
Yeah.
So yeah, no, I haven't done anything.
Okay. All right. Well, thanks. All right, so let's get started. So last week we talked about the BAAI worm paper, and I went back to the archives of the YouTube channel, and I found that there was this project called MetaWorm, which was their preprint of the paper. So they had that same group at a preprint, and it was this different name that they gave it, and this was the paper where they had talked about the different tools, and they talked about OpenWorm, and I guess they used some of the OpenWorm tools in the revision, the BAAI worm revision, and so that's what we covered last week, and we went over their model and what they did, the multi-compartments of the brain and then the body environment and all that. So I just wanted to give sort of a little bit more context for that. He also had a comment about some other things on YouTube. I'll have to bring them up at some point. Okay. Thank you, Soham. So that's what's going on there. Let me go back, and today I think we're going to go over 2 papers, maybe a little bit about one and maybe focus on another. So this one here is Diverging Network Architecture of the C. elegans Connectome and Signaling Network. This is a paper by some network scientists who are doing, I think they're all network scientists. They're doing neuroscience and network science. and they're going to be talking about two different types of C. elegans connectome. One is a connectome based on, I guess, like synaptic connections, and the other based on signaling between cells. And of course, we've talked about networks in like embryo networks where you have cells that aren't neurons. And one of the connectivity criterion for that can be some sort of signaling between the cells. So you have all sorts of signaling pathways and extracellular signaling pathways that affect, where one cell can affect another cell. You have endocrine signaling, you have juxtacrine signaling, and all these other types of signals that can be sent between cells. And so their distance determines how well they can receive those cells. And so that's happening in the connectome at the same time that you have this chemical connectivity through synapses, and of course the physical electrical connectivity through gap junctions. So in this paper, they're going to talk about some of these, what they call diverging network architectures. So you'll have basically the same set of cells, but two different connectivity criterion to distinguish those separate sort of parallel networks. And so sometimes they're going to have the same connections, and sometimes they're going to have different connections. And so let's go to the abstract here. So they start by saying that the connectome describes the complete set of synaptic contacts through which neurons communicate. While the architecture of the C. elegans connectome has been extensively characterized, much less is known about the organization of causal signaling networks arising from functional interactions between neurons. So, people have determined, of course, in C. elegans, you can do this, determine the full connectome, every neuron and their connections, in different ways, but we don't necessarily know about the signaling networks that cause things and the interactions between neurons that result from those signaling networks. You get signaling networks that produce things that affect the synapse and the things in the gap junction, but you don't have a mapping of those like messenger networks outside the cell. So that's what they're trying to do here. Understanding how effective communication pathways relate to or diverge from the underlying structure is a central question in neuroscience. Here we analyze the modular architecture of the C. elegans signal propagation network, measured via calcium imaging and optogenetics, and compare it to the underlying anatomical layering measured by electron microscopy. So they're going to be doing this, they're going to be looking at the signal propagation network through calcium imaging and optogenetics, and then they're going to look at this anatomical layering by electron microscopy. So they're going to have these two different measures of how to do this. Compared to the connectome, we find that the signaling modules are not aligned with the modular boundaries of the anatomical network, highlighting an instance where function deviates from structure. An exception to this is the pharynx, which is delineated into a separate community in both anatomy and signaling. We analyze the cellular compositions of signaling architecture and find that it is modules that are enriched for specific cell types and functions, suggesting that the network modules are neurobiologically relevant. Lastly, we identify a rich club of hub neurons in the signaling network. And then this is this rich club that people have identified in the C. elegans connectome. These are neurons that are highly connected to other neurons, and they form this sort of set of hubs that do a lot of the heavy lifting in the connectome. So you get cells that are, key interneurons that process a lot of information between different parts of the neural network in the C. elegans. So, it's like a small subset of neurons that perform these outsized roles. You see the same thing in these signaling networks. You see rich clubs of hub neurons that receive signals and that respond in kind. And those networks, even though they have a different topology or different connectivity, they still and I don't know in the paper if they identify them as the same neurons, but basically they have this rich club as well in this kind of a network. The membership of the signaling rich club differs from the rich club detected in the anatomical network, challenging the view that structural hubs occupy positions of influence in functional networks. So this is, okay, so they actually say that in this case, the signaling rich club differs from the rich club detected in the anatomical network, challenging the view that structural hubs occupy positions of influence in functional signaling networks. So this is where you get this divergence again between the two networks. Our results provide new insight into the interplay between brain structure in the form of a complete synaptic level connectome and brain function in the form of a system-wide causal signal propagation atlas. Okay, so that's what they're doing here. So basically, to recap, you have these networks that are the same set of nodes, and you're using two different criterion to characterize them. One, which is this sort of structural characterization, in this case with the connectome, and one which is functional, which is the signaling network. And you see that they have, even though they have the same neurons, and the neurons have a connectivity, which is functional at the level of the synapses and the gap junctions, that the signaling network is going to have a different topology, and that you can have, like, if we think about the connectome, we think about it as being connected in the way that we can sort of identify the connections between cells, and then that's all the connectivity they have. But in this case, what they're saying is that there's this signaling layer which has a different sort of structure of connectivity, and it doesn't necessarily overlap with what's going on in the, say, synaptic or gap junction mediated connectome. Okay, so they basically say how the structure of a biological system supports its functional repertoire is a central question in biology. A defining feature of this question is that it seeks to relate two lenses into the same biological system. So what they mean is that you want to be able to see if there's this, set of functions in a biological system, can we uncover all of them, and are they all aligned in the same direction? Basically taking two lenses into the same biological system through these different systems, different measurements of these systems, and looking at them and saying, are they sort of consistent or not? In neuroscience, the relevant structural lens is the network map of synaptic connections in the brain, called a connectome. And so that's what we think of when we think about the wiring of the nervous system. The functional lens is given by network maps that report how the activity of one neuron in the brain either relates to or influences the activity of others. These can be measured by functional MRI, electrophysiology, or calcium imaging. Network science provides a powerful toolkit to characterize and distill key properties of these networks. There has been an implicit hypothesis that since network properties capture fundamental features of the brain, they should persist across both of these lenses. So again, we want to have this consistency, but we often don't get it. However, in studies of humans, there is evidence that network properties can differ, sometimes dramatically, between function and structure, including measures of centrality and community structure. So this is where this connectivity and this consistency of connectivity becomes important. Because in the network, you're looking at the nodes, you're looking at how they're connected, and those connections, direct or indirect, will have an effect on different network indicators or measures. Centrality, of course, being how central a node is to the network. And community structure is how tightly nodes are connected to one another, sort of locally, or if they're able to form discrete subsystems like modules or other types of things. And it's a hard thing sometimes to detect communities and networks, because you often have to use algorithms to say whether this one subset of nodes is more connected than any other subset of nodes, and whether that has some implication for functional coherence. So in other words, if we're looking at a subsystem that we suspect is involved in feeding behaviors, we might look at a number of neurons that are candidate neurons and then look at how tightly they're connected in a network setting. And if we find that they form this community, we might say, okay, there's a community or a module in the C. elegans nervous system for feeding. So this is why this is important. But of course, if we have these two different criterion for defining a network, those community structure can be even harder to sort of implicate in any kind of behavior or any kind of functional consequence. So they give some citations for these measures here, and this is basic network theory, network science stuff, but I think it's relevant here because it has, I think we need to think more deeply about these kinds of measures and how, depending on how we're using our networks or implementing our networks, how they are measured and what they mean. Okay. So, therefore, to the extent of which properties of the brain structural network match properties of its functional network, this remains unclear, especially at the brain scale and cellular level. So, if we have these two overlapping but diverging networks, are they giving us, the same signals? Are they really telling us as much as we think they are about the functional networks and the structural networks that we're deriving at any one scale? So that's basically it. And, in previous work, we've introduced this concept of diverging integration or divergent integration, which is where in the embryo you have these cells that differentiate into neurons, into somatic cells, and they diverge in terms of their function, and they form different networks, but they're networks that are connected at some key point. So if you have a bunch of somatic cells and neurons that are sort of neighbors in embryogenesis, and then they diverge, they migrate a little bit, and then they change their fate to different, form different networks as they become adults. Those cells diverge in terms of their functionality, they diverge in terms of their network connectivity, but they remain connected because they're part of the same organism. So they're kind of hitting on this theme as well. And it's interesting because we often think of networks as static in the sense of, how we're defining them. We say, okay, this is a connectome, this is what it looks like, it's, that's what it is. But in fact, connectomes have their origins in different types of cells. They have different functional attributes, which make them harder to understand. Okay, so they talk about how we can measure the connectome. We can measure it through sort of these synaptic connections and through gap junctions. We also need to look at gene expression, neuromodulators. We need to look at functional connections in the absence of wiring, like extrasynaptic signaling. So we have these peptidergic, actually we do have a peptidergic connectome that exists. And that peptidergic connectome is where you have peptidergic signaling or signaling via peptides. And peptides are a lot less, they're a lot more promiscuous than synaptic transmission. So you get these peptides that are released and they diffuse through the extracellular milieu to activate other neurons. So you can have these polysynaptic pathways that are activated through these peptidergic connections. So that's another kind of example of sort of going beyond your typical set of synaptic connections or gap junctions to look at how signaling affects, a greater portion of the network and actually changes how we understand the connectivity. But from my understanding, the peptidergic connectome is pretty consistent with the regular connectome. And so, there are all these different mechanisms that we can look to measure to get some nuance on what exactly the connectome is doing and what exactly it looks like. Okay, so but this also makes it challenging to translate between the networks of structure and function. So that gets into their work here. They investigate and compare the properties of both networks, wiring and signaling, focusing on their community structure and rich club properties. So what they're looking at is they're looking at the connectome. While the connectome encodes only direct synaptic connections between neurons, the signaling network captures effective connections that arise from the combined effect of not only direct connections, but also all interactions that span multiple hops of the anatomical network or polysynaptic chains. This is also true of extrasynaptic signaling that is not constrained to flow along synapses. So they did this where they considered binarized direct connectome and signaling networks of the same head neurons. So they took each neuron and they considered, or actually they took 188 neurons from the connectome and they considered them head neurons. And so from this subset of 188 head neurons, they considered them to have sort of, they started with the same head neurons that had different numbers of edges. So they started with sort of a root of the network, and then they sort of reconfigured it according to kind of different criteria. Thus, the networks differed in terms of their density and their distribution of motifs. So they had these different types of networks that they started. They started with head neurons, and then they considered these binarized directed connectome and signaling networks, and they ended up with a bunch of alternate networks that gave different numbers of edges. So they had 1,151 edges for the signaling network and 3,250 edges for the connectome. So that just means how many connections there are between neurons, starting at these head neurons and moving down. And so the signaling network was much sparser than the connectome, from what I understand from this. So that means that these networks are different in terms of their density. The signaling network, or the density of the signaling network, is much lower than the density of the anatomical network, and then the distribution motifs is different as well.
I got a question.
Yeah.
Is the smaller network a subset of the larger network?
Well, it's in terms of the nodes, yes. Yeah.
Okay.
Yeah.
Okay, go ahead.
Yeah.
Thanks.
Okay. So the clustering coefficient is also lower in the signaling network. It's a bit less than half of the anatomical network's clustering coefficient, as we might expect. We also know that C. elegans connectome, through connections, what we call the anatomical network, is a small world network, which is where you have this path length across the network, which is relatively short, given the amount of connectivity you have, given the number of nodes. And so the small world network is where you can take any one node or neuron and get to any other neuron in a relatively small number of hops. And so this is something that we also find in the signaling network. It's a small world network. And so we find this for these head networks. So both of these types of networks are small world, but they have different properties in their connectivity. Okay, so they talk about the neural networks being hierarchically subdivided into subnetworks. A lot of times people will call these modules or communities or clusters. This is, of course, important for function, but it's a key hallmark of structure. This provides a balance, but of course we know that these subnetworks aren't isolated. They have connections between them. This is why we can observe the small world behavior. We can observe these different properties of networks that, where you can go from one end of the network or one end of the whole network to the other relatively in a short path length, because we have key connections between sub-networks. This provides a balance between segregation and specialized functional units. and integration of information across distant neural elements, and therefore informs how the nervous system performs computations. So they tested whether the anatomical and signaling descriptions of the C. elegans brain shared a similar module structure. We used a hierarchical variant of the stochastic block model to independently subdivide the wiring and signaling networks into nested communities. A stochastic block model is just a sampling technique that people often use to artificially subdivide a network and test out different structural motifs and structural instances to look at their function, look at their structure. So they use this kind of approach, the block model approach. We found stark differences in the modular organization of these two views into the C. elegans brain. The anatomical connectome was clustered into 20 communities in the first hierarchical level, with the smallest containing only one neuron and the largest containing 20. So this is where you have these different communities. You can get from like a community of one neuron, which isn't really that interesting, to communities of 20, which is, maybe more functionally satisfying. In contrast, the signal propagation network was optimally partitioned. And so this is figure 1. This shows the hierarchical modular structure of the C. elegans signaling, propagation, and anatomical network. So this shows the anatomical network in A, and this shows a signaling network in B. So you can see that they use this sort of circular layout for their network. They have these different subnetworks here that are numbered. I guess these are the communities. Let's see what they say in the, sorted by community assignment. So in this case, they have from 1 to 20. So you can see on the chart here that they go around. There's connectivity across communities. There's connectivity within the communities, of course. So this just shows the structure and how it differs in terms of connecting across communities. So for the anatomical network, we have this matrix, which is postsynaptic versus presynaptic. Basically, the presynaptic communities connect to the postsynaptic communities at some density, and you can see that some are more highly connected than others. You can see that in the circular plot as well. And for the signaling networks, you see it's much more sparse. We knew that from the statistics. We can see that in the circular plot that it's much less dense in terms of connectivity. You still get this sort of connectivity between communities, and you also get this in the matrix as well, where we see communities 5 to 5 and 1 to 1 are very densely connected, and the rest, are varying degrees of sparsity. So this is interesting, just thinking about like the differences between these networks and what that might mean for function. So they give a list of communities here in the signaling network, what's in each community. So we have a lot of the I2 and I3 and I4 IL2 neurons, the M1, M2, and M3 neurons. We have some of the NSM neurons and the URYVR neuron. So that's in community 1. Community 2 has URYVL, URYDL, IL2DR. So you start to get like things that are sort of similar to what's in community 1. Sometimes it's the bilateral pair, but most of the time they're, you don't see too many bilateral pairs in the same community, which is interesting. You see ILR and I1L and I1R in the same community, and 2. You see IL1DL and IL1DR in the same community. So you see bilateral pairs in community 2, but not in community 1 so much. Community 3, you see some asymmetry in terms of the lateral pairs. AFD is only the left one, AIM is only the left one, AIY is only the left one. AIY has the bilateral pair. but ASJ only has the left one, and so on and so forth. So you sometimes see these bilateral pairs in the same community, sometimes they're not, which suggests something interesting from a functional standpoint. If we went to the anatomical connectome, we would see that sometimes the left neuron is doing something a little bit different from what the right neuron is doing. Sometimes if you look at like when they differentiate in development, the left one differentiates before the right one. At least it's as detected through the methods that we have. So there are these asymmetries that we know that exist in the connectome in terms of when a cell originates, how it's wired, and then also apparently the signaling relationships between them. So they have this part on the distribution of bilateral pairs across communities. So a majority of the C. elegans neurons consist of bilateral asymmetric pairs that are often connected to each other via gap junctions and tend to have similar gene expression, wiring, and correlated activity patterns. So we expect these bilateral pairs, since they come usually from the same mother cell in development, and they have the same nomenclature. They usually have the same type of functional role on either side of the worm, but they have differences in terms of their behavior. But we want to look to these kinds of pairs because they seem to be the most similar neurons in each part of the connectome, right versus left. So we have this test that we can do, which is to look at the bilateral asymmetric neurons and see if they're in these same communities, or if they're doing the same things in the network, or if they're doing different things in the network. As we would expect, bilateral asymmetric neurons are much more likely to be found in the same communities that are across different communities. So they actually say that they find, if you go systematically through this, bilateral asymmetric neurons are much more likely to be found in the same communities, and this is in the anatomical networks. Much more likely to be found in the same communities than across different communities for both the anatomical and signaling networks. In the anatomical network, bilateral neuron pairs were 38 times more likely to be in the same community than in different communities, and in the signaling network, they were twice as likely, which means that there's a difference. We were looking at the signaling networks. We saw that sometimes we had pairs that were not symmetrical in the sense of being in the same community. So the signaling network, maybe by virtue of being less dense, being more sparse, but maybe also because it's more promiscuous in terms of the signal, that it is less likely, but also twice. It's actually okay. So they're twice as likely as just random neurons to be in the same community, but they're also much less likely than anatomical networks to have those bilateral pairs be in the same community. So it's a little confusing, but basically bilateral neuron pairs are almost always in the same community, 38 times more than like random cells. If you were to take any two cells that weren't of the same sort of bilateral pairing, then it would be 38 times more likely. In the signaling networks, they're twice as likely. They're using that same criterion, but that's much less, of course, than the anatomical network. This finding is consistent with the hypothesis that some features should be conserved across both network descriptions. So we should have this conservation across network descriptions, but it's going to vary because of the nature of these networks. Okay. So they give, okay. So we'll switch gears here to finish up this paper by talking about how communities are enriched for neuron type and role. Clustering of anatomical networks has previously been used to identify communities of neurons that are enriched for cell types or known functional cellular roles, including Drosophila and C. elegans. So we've seen this in different organisms, in these model organisms where we know what the cell types are. We can build a model with a connectome pretty easily across individuals. We have these archetype connectomes. Consistent with prior findings, our module clustering of the C. elegans anatomical network recapitulates anatomical communities that are each significantly enriched for no more than one of the three C. elegans neuron type classes: sensory, inter, and motor neurons. So what they're looking for here is they're looking for these rich club networks. Similarly, they're each enriched for no more than one of 5 predefined cell roles. And so they give this figure here, which is figure 3. Communities are enriched for neuron type and role. So they show this example here where they have, these are their 6 communities for the signaling network. They show that for sensory, inter, and motor neurons, you have these different memberships in the signaling communities, and you see that the z-score is variable across here. For community 2, you have a z-score of 4 for sensory neurons, and negative z-score for inter and motor neurons. In community 1, you have a high z-score for motor neurons and a low z-score for sensory and inter neurons. Community 3 has a lot of motor neurons, 4 has interneurons, and then 5 has almost a 0 z-score for all 3. And then I guess that suggests that it's well mixed in that sense. And then moderate z-score for sensory neurons in community 6. Then they have this for other, like they break out the neuron types a little bit more finely. Pharyngeal neurons, amphit, sensory, locomotory, are just interneurons, VNC, motor neurons, ring interneurons, and all others. And they show this similar variation across communities, showing that there's, I guess what this means is that there are a greater number of different types of neurons in certain communities over others. Some communities specialize in certain cell types, whereas other communities specialize in other cell types, which is what you might expect to find if you're using a community detection method and you're trying to find like communities that are sort of do basically the same types of things, or expect a mapping between function and structure. So if you have a lot of structural similarities between a set of neurons, you might expect functional similarities as well. And so this shows this for the anatomical networks. You see the same sort of pattern. Of course, we have different communities here, so we're going to have some communities that have more of an emphasis towards sensory neurons, some towards interneurons, and some towards motor neurons. And this holds true for this breakout of function as well. So for pharyngeal, for amphit, sensory, locomotory, inter, VNC, motor, ring, inter, and all other, you see the same pattern. Okay, so this is getting us to this rich club of neurons, and to get to the rich club of neurons, we have to understand sort of these community interactions. So we have assortative, disassortative, and core periphery interactions. And so the assortative are where you have neurons that sort of assort by some criterion, where they, maybe join some highly connected cell, where they join like cells in a community, and this basically serves the purpose of sorting the network. So the network connectivity is defined by sort of like cells or by cells that have a high connectivity, and it sorts the network in a way has this sort of natural structure like this. In a disassortative sort of organization, you have cells that connect to just random cells, and you don't really have very much structure that results from that. The core periphery model is where you have this very strong signal for connecting to core or these rich club neurons, and that's where you have these green stars surrounded by these network nodes that are peripheral. So the peripheral nodes will much more likely connect to these starred neurons, or these core neurons, which then end up being these rich club neurons. And you form this strong core periphery structure. And so you see in both in terms of signal propagation and anatomical networks that you have a similar sort of breakdown of connectivity types. So in signal propagation, or when we form our network based on signal propagation, the assortative network motif is found in 68% of the network. The core of the network involves 14% of neurons that are connected together. And then these peripheral neurons consist of 18% of the structure. So this core periphery structure forms about 32% of the network connectivity, and the assortative part forms 68%. In terms of anatomy, we have the core and periphery drop to a combined percentage of about 19%. You have this 4% disassortative connectivity motif, and then for assortative, you have 77%. So this is both because the network is less, it's more sparse, and it's also more promiscuous in terms of its connectivity. You see a small amount of disassortative connectivity, but you see a greater amount of assortative connectivity as opposed to this core periphery connectivity, which is indicative of these rich club neurons. So I think that's all for that paper. I just wanted to really kind of give a strong review of that paper and kind of go deeply into it to show how we might look at network connectivity for different types of connectivity criterion and what that means for what we think of as the connectome. Okay, looks like we lost Mehul and Soham, but we'll press on. So this second paper, and we'll go over this more quickly, is this paper, WormID-Benchmark. So this is extracting whole brain neural dynamics of C. elegans at the neuron resolution. Okay, so the abstract of this paper is the nematode C. elegans is a well-studied model organism for characterizing the structure, connectivity, and function of a complete nervous system. So we, again, like in the last paper, we can use different types of methods to extract different types of information from any sort of thing from the biology we want, within the scope of, so we can extract connectomes that are, these chemical or physical connections. We can extract connectomes that are peptidergic. We can extract them that are, different types of signaling modes. We can do a lot of things like that. We have a lot of new methods in 3D light microscopy and fluorescent protein tagging that have brought us closer to capturing the neural dynamics of the worm at whole brain resolution. Nevertheless, capturing a complete map of neural dynamics using these high-resolution recordings requires solving 3 specific challenges. So, this is not without its issues. I mean, they've applied these methods and gotten data, but there are 3 challenges that really probably still need to be overcome. The first is detection of individual neurons in fluorescence videos, and we know that from our own work with trying to segment cells, follow the fluorescent signal, make sure that the fluorescent signal isn't spurious, et cetera, et cetera. 2, identification of those neurons according to their anatomically defined classes. In other words, how do you map what we know from, say, like the light microscopy, which defined the really early studies in C. elegans, to some of these other methods where you have to track the cell. We have to know what cell is this specific cell type and follow it across different individual worms and across different individual observations. And 3, tracking of neural positions over time. So when we get our Connectome, we want to be able to track especially if we have a behaving worm, those neural positions are going to change. We're going to want to see, across function and across development especially. Successfully addressing these challenges with high sensitivity, specificity, and throughput can enable us to analyze a large population sample, providing unprecedented insights into the structure and function of an entire brain at single neuron resolution. So we have not been able to do this really in any other model organism, maybe because C. elegans is such a fabulous model organism, but also because, this is I guess this is probably something where we have a decent sized connectome along with the ability to do this, to know what the cells are and to track things accordingly in culture. To facilitate the scientific goal, we have curated publicly available annotated data sets from 118 worms across 5 distinct laboratories and established systematic benchmarks, decomposing the overarching objective into 3 well-defined tasks. So those are neural detection, neural identification, and spatiotemporal tracking. So looking at these things in time and in space as they move, the organisms move, sometimes the cells migrate. Identification, which is where you want to correctly identify the cell, and then detecting something that is a neuron versus not a neuron. Our preliminary analysis has revealed considerable room for improvement in existing state-of-the-art computational methods. Consequently, we envision that our WormID-Benchmark can catalyze efforts by a broad audience specializing in computer vision to develop robust and accurate methods that significantly enhance the throughput of generating annotated whole brain neural dynamics datasets. We make our benchmark results reproducible. Our code is available on GitHub. This is the GitHub repository here. This is WormID. All right, so let's go through the repository. So this repository contains the code necessary to reproduce the results and analysis in the WormID. This is from NeurIPS 2024, and this is the benchmark paper. I think this paper was published on bioRxiv. Yeah, so this was actually a NeurIPS paper in 2024. To clone this repository with all included submodules, run git clone recurse submodules in the command line, or run git submodule update to selectively clone specific submodules. So they have different sorts of tasks that you can do to run this. The first is neuron center detection. So the first benchmark is where they'll take a neuropal image as input and predict the location of each neuron. We evaluated 2 classical models for cell segmentation tasks. There's CellPose and MicroSam. So SAM is the segment anything model. So they're actually, we talked about CellPose and MicroSam from the Google Summer of Code, period, where we're trying to find the right algorithm to do segmentation. They're using 2 popular, biological segmentation algorithms here. So they're going to use these 2 classical models. You download the data, you pre-process it, you run inference.py and evaluate.py, which should be in the repository. And then you run those for each individual model. Then you aggregate the results of the evaluate.py and the neuron detection datasets. So that's the first one, and you're detecting the center of a neuron with this method. Task 2 is neuron-pal neuron identification. That's where we have our ID benchmark notebook, and this contains the code for running the analyses presented in the benchmark paper once the results have been produced for each model. So they look at this technique, coherent point drift, and they implement coherent point drift using this paper here, which is introducing this method. This code can be found in the FDNC neuron ID submodule, which is up in this part of I think this one. These are all empty for purposes of this repository. You have to clone the repository, and then that's going to be a working repository for that test. So this is for this coherent point drift test. To reproduce the benchmarking results, use the run CPD notebook script to generate the results CSV files. Relative data paths are in the run CPD notebook file. These paths assume that the data folder is at the root level of this repository. They may need to be changed if the data is in another place in your local file system. Statistical analysis. The code for this analysis can be found in the NWB elegans repository, presented in Sprague et al. This is this bioRxiv paper. Further details on the model itself can be found in the original Statistical Atlas paper, which is shown here. And this is this script contains the code for retraining the Statistical Atlas and generating the benchmark results. So this atlas here is the Statistical Atlas of C. elegans neurons, and it uses NeuroPal, which is a means for assigning the correct identities to all neurons in the worm via deterministic fluorescent color map. So this is a NeuroPal. This is a common way to identify cells. And the idea is to sort of retrain the Statistical Atlas on your data and generate those results and see how well they do. Then there's CRFID. So the MATLAB code for CRFID is provided as a submodule. This method is found in this paper on eLife, which is this graphical model framework for automated annotation of cell identities and dense cellular images. CRF stands for conditional random fields. This is a probabilistic graphical model framework that allows us to do unbiased and automated cell identification. So they test this on C. elegans in this paper, but they reproduce it in this using this GitHub repository. Okay, and then finally, task 3 is neuron tracking and whole brain calcium imaging. So in this task, we will take calcium video data, or CGAMP, and the ground truth neuron locations at the reference frame as input, so they have a ground truth for these neuron locations in this movie data. They take the first frame, and then in subsequent frames, they predict the location of each neuron. We evaluate 2 classical models for cell tracking tasks, ULTRAC and 3D Cell Tracker. Detailed instructions are included in the individual folder of each tracking method. Okay. So they want to be able to allow us to verify different tracking methods, make sure that they're, robust. So they talk about ULTRAC for a minute here. The segmentation part in ULTRAC can be performed using watershed, cell poster, Stardust types of segmentation. Here we use Stardust with the pre-trained weights available at OSF. So it's this OSF repository here. Pre-trained and fine-tuned weights can also be downloaded from Google Drive. And so they give instructions on how to run the pre-trained version of ULTRAC and the fine-tuned version. So that's a little bit about that paper and its associated GitHub repository. Hopefully if people are interested in Google Summer of Code, they can check that out. Maybe that will help you in your Google Summer of Code work, especially in, getting some data sets and verifying the data sets and performing benchmarks that you can then report. Okay, it looks like Dick had to go to another meeting. So any questions about any of the papers that we talked about? You're muted, Susan.
It's just myself. So,
okay.
I'm interested in the physical connectome. I need to get into networks perhaps a little more because it relates to the tensegrities I was doing. And the network nodes are kind of like the nodes in the tensegrity, I believe.
Yeah, similar.
And as far as the signal connectome, that would depend on the connections between the dendrites and the and the axons, wherever they connect, wouldn't it be? Like,
Yeah, so for like a typical, well, there's of course the chemical connectome, which is where you have synapses between two cells. Where you have a cell, a neuron, and an axon, and then those lead to the synapses and dendrites, which have some form of chemical communication. So you have some sort of neurotransmitter or peptide there. And then you have the gap junctions, which are the electrical connections, which are physical between cells. There's an exchange of neuropeptide there sometimes, or of neurotransmitter there. And then sometimes you have these peptidergic connectomes, which are where peptides will leak out across like in extracellular space, and will be taken up by neighboring neurons. They may have synaptic connections, they may not, or gap junction connections, or may not.
okay, it's just that I've heard of, I don't know, dendrites connecting to the middle of an axon sometimes. So you get
Sometimes, yeah, it's
you get that happening too. I guess well, you still get gap junctions.
yeah, you do. Well, gap junctions are usually, those connectomes are different from the chemical connectomes of synapses because
they're not a synapse. They're,
right. It's a different
gap
motive.
junction. Yeah, which are more relevant in a heart, maybe.
Yeah. And then, of course, in non-neuronal cells, you have gap junctions, and they perform communicative things. And you have, peptides that diffuse, or other types of signaling molecules that diffuse in non-neuronal cells that don't, there are no synaptic connections there, but they do have this ability to communicate by signaling, and that's, of course, this signaling network. So you have some parallels in non-neuronal cells, but not the chemical connections that you find in neurons. And so they're also, you also have different types of ion channels in non-neuronal cells that allow for this sort of, we think of neuronal cells as having ion channels and expressing ion channels, and that's what makes sort of the ability for chemical communication. But we have this in non-neuronal cells as well. That's a lot of Michael Levin's work on bioelectricity, where you have ion channels that produce electrical signals that aren't anything like action potentials, but can perform these communicative things between cells. So it's really there's a lot of connectivity between cells, depending on the cell type. There are a lot of modes of communication and connectivity, and it's really, I like this idea of divergent networks, because even in the same group of cells, you have many different types of networks, but especially across cells of different types, you have these networks that join together at key points, and they're all kind of connected.
Okay.
Yeah.
So mechanical connection still needs to be considered.
Oh, of course, yeah, and then of course you have your biotensegrity structures, which are where they have this mechanical connectivity that is keeping everything together.
Yeah, it's sort of instantaneous as well. You push one thing and it moves the other.
Yeah. Okay.
At least I can show that on my small tensegrities I have here.
Yeah.
That if you push one thing and it changes the other side of the structure.
Yeah.
It's kind of neat. Anyways.
All right. Well, thanks for attending.
Oh, well, I look forward to it every Monday.
Yeah.
This starts off my week, so.
Yeah. All right. Have a good week.
All right, thank you.
Thanks. Bye.
Bye.